All right, welcome everyone. Hello, welcome to the Signals Notebook Quarterly Connect community series. My name is Zev Wasatsky, and I am the Senior Principal Marketing Manager for Biologics at Revvity Signals, and I will be your moderator for this session. This is an ongoing Signals Notebook community series, and it will be an opportunity for you to learn about newly added Signals Notebook functionality and hear how others are using Signals Notebook to improve productivity and data transparency through enhanced collaboration. This Signals Quarterly Connect will focus on hierarchical editing language for macromolecules, or HELM. Our previous sessions have been recorded and are available for streaming at Signals Notebook Quarterly Connect. And the one thing to mention here is future dates for Signals Quarterly Connect for 2023 have been shared along with this registration. So I hope you will join us in the future programs scheduled for November 29th in this coming year. Speaking of upcoming dates, for our user conference, Nexus, please join us virtually and in person on these dates in October. We will drop the link for more information within the chat. And before we begin, a couple housekeeping reminders. We will have ample time at the end for questions, so please submit your questions through the question box located on the right-hand side of your screen. And again, this webinar is being recorded, and the recording will be shared with you, with all the attendees, in the upcoming days. So let's jump into the agenda for today. Okay, so first we're going to be introducing HELM and providing an overview and history of how and why it came to be and the players along the way. We will then be discussing HELM functionality within ChemDraw, integration of HELM into Signals notebook workflows, bio-related molecule capabilities. We'll see HELM in its collaborative workflows using HELM, seeing HELM in actions. And then finally, we'll summarize and have time for questions. Okay, so before I introduce our speakers and demonstrations, let's all get on the same page on what is HELM, why was HELM made, and where it came from. So again, hierarchical editing language for macromolecules, or HELM, is a way to represent the molecular continuum from atoms to complex polymers and to standardize how they are represented. Communication along this line is extremely important to be effective. So the main challenge in creating these complex molecules is there wasn't a standard way to describe from an atom or a simple polymer and any modifications that went along those. So in steps HELM. The goal for HELM is to enable the representation, the registration, storage, analysis, and visualization of an ever-growing set of complex biomolecular structures. The notation language has to be simple, but versatile enough to represent all of these types of structures. And most importantly, these notations have to be easily generated and parsed by computer programs. So from a historical perspective, scientists at Pfizer first developed this technology, and through the Pistoia Alliance, which is through many companies, including but not limited to BMS, GSK,
Revvity Signals,
and Roche, brought an evolved HELM to the community and where it is today, which is now an IUPAC standard. And I think this is quite an important point because it showcases why it is increasingly critical for tools to provide the ability to model HELM and support its functionality in an intuitive way. So with that, I'll introduce our two speakers. Our first speaker today is Dr. Lindsay Rickershauser, product manager for ChemDraw. Prior to this, she graduated with her PhD in organic chemistry from Johns Hopkins. She went to do bench science in the synthesis of stable label isotope molecules. She then turned her focus to cheminformatics and software development, and has helped to develop and commercialize and launch leading retrosynthesis software prior to our current role. Thank you for joining us today, Lindsay. I will turn it over to you to discuss HELM capabilities in ChemDraw, integrations with Signals Notebook, and custom HELM libraries.
Excellent. All right, thank you, Zeb, for the kind introduction, and thank you everybody for joining today. It's great to have you here. So I first actually wanted to talk today about our current HELM capabilities that are available in ChemDraw Desktop. So one thing that actually may surprise you is that the representation of biopolymers and using HELM has been available in ChemDraw Desktop since 2017. Since then, there have been significant improvements and enhancements in each major release. And as you'll see today, we've brought much, if not all, of that capability into Signals and actually begun to improve upon it as well. So what are our current capabilities? So first of all, you can build and draw sequences using a HELM editor tool in ChemDraw Desktop. So you can draw things like peptides, oligonucleotides, and any conjugates you may want with chemical linkers. You have full copy as and paste as support. So if you have a FASTA string or a HELM string, you can paste that directly into a ChemDraw canvas and render that sequence, provided that you have the monomers available in your monomer libraries. That brings me to monomer management. So you do have standard libraries available to you in ChemDraw Desktop for natural monomer libraries, the libraries from the Pistoia Alliance, etc. But you also do have the ability to curate your own custom building blocks and monomers as well. And you can even share those custom libraries and centralize them within your organization. You can expand and contract any of these labels to get the full structural representation behind the sequence that you actually build on the canvas using these tools. And then maybe the best and most significant part of this is when you draw these in ChemDraw, the actual document that you save, the CDX or CDXML document, is fully portable, meaning that all of the monomer definitions that you use to build that sequence are actually defined in that document, making them portable and renderable without the library that it was actually built from. So you can share it with internal and external colleagues that may or may not have those custom monomers that were used in that sequence. So any sequence that's drawn on the canvas can actually be represented with labels, or the structures expanded, making it quite easy to make things like chemical modifications or cross-links within the sequences where you're desired, using those editing tools that are available in ChemDraw. However, because of the difficulty of actually distinguishing between the different polymer types like peptides or oligonucleotides or the chemical linkers, just from that text-based format, we actually have created an additional sort of style that was part of the 22 release of ChemDraw that allows ChemDraw to now render sequences in a graphical representation. And so this allows this peptide oligonucleotide conjugate that you see on the screen to go from this text-based view to a nice new colorful and graphical view. So what you'll notice immediately is you have some quick visual indications in terms of what the polymer types are. So you have different shapes according to whether you are drawing a peptide, a chemical linker, or an oligonucleotide. So you have unique shapes for all the sugars, phosphates, and nucleobases, and everything that you're drawing with here, which allows you to quickly identify exactly what you're looking at. In addition, you have color coding according to the natural monomer that these monomers are built from. So where you have custom or unnatural monomers that you're building and working with, you have color coding back to the natural monomer, so you can easily identify where that's coming from, what the origin is, and you can quickly sort of identify it within that sequence. So you may have actually seen this graphic representation in Signals already. It's been around for just a little while. However, what's actually been missing until very recently is the ability to actually build the new biopolymers in Signals Notebook itself. So since the 22 release of ChemDraw Desktop, we've actually turned our focus to bringing the Helm editing functionality into ChemDraw JS, which is the ChemDraw component used in Signals Notebook. And so I'm happy to say that what I'm about to show you and talk about today is actually now in beta in Signals Notebook. So this editor has new and improved functionalities as compared to ChemDraw Desktop Helm editor. However, is built upon all the Helm capabilities that actually already exist in ChemDraw Desktop today. So the new editor can be invoked by a tool on the toolbar. You can, same as desktop, draw things like peptides, oligonucleotides, conjugates. You have full copy as and paste as support, but we also have brought new enhanced features into this tool that are exclusively available here in Signals. So that includes things like being able to favorite monomers. So where you're using specific monomers quite often and they might be complicated or hard to find in large libraries, you can now favorite them and collect and curate them in a specific tab in the editor itself. You also have a text input. So if you want to quickly type out a FASTA string, you can type that into our text tab, insert it onto the canvas, and start making quick point edits rather quickly and easily from that. You can also paste a Helm string into there as well. We've added a little bit of increased flexibility in terms of editing a sequence, so when you're inserting or replacing monomers. And then we also have a quick and easy ability to actually look at the structural information behind any of those graphical monomers within ChemDraw JS. So where you're not sure what a particular symbol actually relates back to in terms of the structural information, you can hover over one of these sequences and get that structural information rather quickly so that you can begin making those edits where you'd like. And so in order to bring Helm fully into Signals, we also needed a way to centralize and manage those monomer libraries. So we actually created the Helm monomer curation application. So this is a new application within Signals that allows administrators the capability of uploading those proprietary monomer libraries for use in the Helm editor in ChemDraw JS. So now you can actually use your own curated libraries to build those sequences in Signals. So whenever these libraries are actually imported and activated on the tenant, users with appropriate roles and security policies will be able to access them anywhere you can invoke that Helm editor in Signals, so in an experiment or a register in materials, etc. The curation application is also now part of that Helm beta functionality as well. Okay, so I wanted to give you guys a few real life examples in demo form of some molecules, how we might draw them and how they can be expressed in this Helm graphic notation. So the first example I'm going to show today is a peptide called terlipressin. So this is a peptide that's FDA approved for the treatment of hepatorenal syndrome. It's a rather small peptide, but when you're looking at that full sort of structural representation, it's a pretty complex small molecule. So if I were drawing that by hand, atom by atom, or even using hotkeys in ChemDraw, that might take me some time to actually get correct. So I want to show you how we can very quickly get that onto the canvas and draw in a rather easy fashion using that Helm editor. The second example I'm going to talk about today is mipomersen. So this is also an FDA-approved drug to treat homozygous familial hypercholesterolemia. It's a mouthful. It's an antisense oligonucleotide that has several modifications. And one of the reasons I want to show this is so I can show how you might make some of these quick point modifications when you're actually building these nucleotides using the Helm editor. So we have methoxyethyl ribose modifications at the beginning and the end of this 20-mer oligonucleotide. We also have a thiophosphate throughout the entire backbone. We have deoxyribose in the middle 10 nucleotides of this sequence, and then we have all the cytidine residues are modified to the 5-methylcytidine. So I'd like to show you guys in real time then how we would actually realistically do this in the HELM editor. So with that, I'm going to go ahead and just jump into signals. Okay, so I'm in my signals notebook and I have these 3 experiments that I've sort of started. So terlipressin is that small peptide that I'd like to start with. So I have that full structural representation actually loaded here for us to see, so we can see the difference once we get the graphic sequence actually built. So before I draw it, what I really want to do is give you just a quick walkthrough of getting the HELM editor open and ready for editing, right? So I have this empty ChemDraw drawing. I'm gonna go ahead and flip it into a full-size document. And then I'm actually going to go ahead and reduce that side panel. So now I have a full screen view to be able to draw this nucleotide. In order to invoke that HELM editor, I just want to select that tool on my toolbar, and now I have that HELM editor panel in the left-hand side of my screen. So what you can see here are all of the different tabs for drawing the different sort of polymer types. I'm right here in my favorites tab, but I can flip right over to my peptide tab to begin drawing if I'd like. One thing you'll notice is you have the library navigation available to you. So if I want to stick with just those natural monomers, and that's all I would really need to draw the terlipressin, I can select that and I'll get all of my natural monomers. And I have some filters here as well if I want to, you know, start filtering for some of those larger libraries. I'll also have this text tab, and so this is actually where I'm going to start. So if I have a FASTA string or a HELM string, I can insert it right here and get it on the canvas rather quickly. And so like I said, this is a rather small peptide, so I can very quickly type in my sequence and get the majority of this structure actually drawn. So I'm going to go ahead and do that now. All right, so that is my sequence, my FASTA string for my terlipressin. I'm going to select FASTA peptide, and I'm going to insert that now on the canvas. So you might have seen that disulfide linkage. So we're not done yet actually in building this molecule. So what I need to do is actually make a cross-link here between these 2 cysteines. And you'll notice I have that hover over capability. So if I don't recognize the symbols, I can go ahead, hover over, I'll get that name, I'll get that structural representation. But what I'm going to do here is I'm going to select the bond tool, and then I'm going to create that cross-link between those 2 cysteines. And the very last thing I need to do is just change that terminal carboxylate into an amide, and I actually now have terlipressin. So I'm just, for posterity's sake, going to go ahead and clone this, and I'm going to open up to that full structural representation just to show you that all of that sort of is still there and behind. So I'm going to open it up. I'm going to ask it to expand the labels, and then I'll do a very quick structure cleanup for you, and you'll see a rather nice representation of terlipressin. So I think that's right about the orientation that we have in our other documents. So this is a really quick and easy way to get this onto the canvas. Of course, like I said, I could come into that peptide tab as well and start drawing using these monomers from my natural monomer library. Okay, I'm gonna now go, so we'll just see the difference sort of in those representations for terlipressin, and then I'll move on to mipomersin. So you saw in the PowerPoint presentation, the mipomersin has lots of different modifications here, right? So I have everything sort of highlighted so you can see all the different modifications that we actually have. So if I were to draw this by hand, this would take quite some time, but with that HELM editor, it actually takes a lot less time than you would expect. So Let's come in here. And now that I have invoked that Helm editor, actually CDJS remembers it. So this Helm editor is now open. I don't have to actually open it from the toolbar to get started if I don't want to. So we walked through the peptide and the text tabs. Now what we actually want to do is start building the oligonucleotides. So I have to use that DNA and RNA tab. So one thing to know about ChemDraw is that it draws those oligonucleotides in full nucleotide form, meaning it draws the ribose base and phosphate all together. So you have some sort of setting up to do when you actually draw these nucleotides. So I'm drawing using the base because that's what I'm going to vary most often when I actually create this oligonucleotide. And so I'll set what I'm going to use what I'm going to draw using my ribose and phosphate. So I have to get to that methoxyethyl ribose. When I actually select this button, I will see the last 5 riboses that I've actually used. But if I don't find what I'm looking for in the list, I can change it and I can, navigate through different libraries. Maybe I want to go into a custom library. I don't have that there, but I can go to the Pistoia Alliance library, and there's my methoxyethyl ribose. If you're not sure what the symbol is that you're looking for, you can also do a text-based search, and it will search according to the name as well as the symbol. Okay, so I've chosen my methoxyethyl ribose, and now I need to get to a thiophosphate. I also have thiophosphate in my last used list, so I'm going to go ahead and select that. But similar to what I just did with ribose, if I didn't see that in the list, I could search for it and I could find it. So now that we're all set up, I can start actually using the editor itself to draw. So the first 5 and the last 5 nucleotides have this methoxyethyl ribose modification, and I'll go ahead and draw my first 5 nucleotides. Okay, so now that I have those on the screen, and just note we've used the 5-methylcytidine in place of the cytidine itself. And so I'll see that color coding, and I will also be able to hover over and see that information if I'd like. So now that I've got my first 5, I need those middle 10 of my 20-mer sequence to be deoxyribose instead of methoxyethyl. So I'm going to go ahead and change that to deoxyribose, and now I'm going to draw those middle 10. And I'm going to just zoom out so that we can see everything as it gets built. All right. Now we have those middle 10 with the deoxyribose. We're ready to go back to the methoxyribose, and we will get those last 5. And let me just do a quick zoom out so that we can see it. And I'm going to just wrap this around in 2 and zoom in so that we can see everything a little bit more clearly. So now I have my 20-mer oligonucleotide. The last thing that I want to do is just get rid of that phosphate. Oops, and now I messed up my, there we go. Oops, oh gosh, darn it. There we go. Let's just do a quick biopolymer cleanup. There we go. Now we can zoom in and see our final thing. So you can leave it in a straight line. You can zoom in and pan. That's easy. Or you can move it into 2 lines to easily sort of see that full sequence if you'd like. It's absolutely up to you in terms of how you do it. Okay, one last thing before I move on to my last example here is adding favorites. So if the methoxyethyl ribose and thiophosphate are a combination that I'm using quite often, I can choose to favorite that so that I always have it very quickly and easily available to me. So what I can do is I can go to add favorites. I've got that methoxyethyl ribose and the thiophosphate. And so I can come in here, I can choose that 5-methylcytidine, my quantity, my thymine, and I can, an adenine, and I can go ahead and select it. what I see immediately is I have little stars. So those are my favorites. I'll see those indicated when I'm looking in this oligonucleotide tab, but I can also flip over to my favorites tab, and now I have those listed for myself, the thiophosphates, that full triplet, so I can quickly and easily get to them. Okay, so my very last example, I'll quickly go through. This is the peptide oligonucleotide conjugate. So these are small, this is a very small peptide and small oligonucleotide. What you'll notice is the oligonucleotide is using a locked ribose instead of ribose or deoxyribose. And I have an alkyne and an azide sort of appendage on each of these so that I can do a click reaction to create that conjugate. So I'm going to come in here and go ahead and build that. So I'll use easy natural monomer library for the peptide. So I'll draw the beginning small peptide there, and then I'll just make a few edits to create that azide. I'm using some hotkeys in the process to create that sort of chemical linkage there. Now what I want to do is I want to draw that oligonucleotide. Like I said, I'm going to use a locked ribose for this. So I'll go ahead and pull down and add my locked ribose, and then I'm going to change back over to natural phosphate. And then I'll go ahead and draw this quick oligonucleotide. Okay, and there we go. So now I need to just add that alkyne, and I'll go ahead and do that. Just a few more hotkeys to get there. All right, now I have my alkyne. So in order to create a reaction with this, what I'm actually going to do is, let's zoom in just a little, add a reaction arrow, and then I'll just go ahead and clone this to below my arrow so I don't have to draw it again. There we go. And I will pull this right over. Now, the last thing that we really need to do is we need to create that triazyl ring and create that connection. So I'm going to build that triazyl. And then I will connect my azide linkage. So all I have to do is now move this over and connect my two carbons, and I've got my product. All right, so there we go. So now I have my reaction, my click reaction, where I've created my peptide oligonucleotide conjugate. So with that, I will go ahead and pass it back to Zev. Thank you everybody for your time and attention.
Great, thank you, Lindsay. We will move on to our second speaker. So for our second speaker, it will be Dr. Larry Mulcahy. He is the Signals Notebook product manager. His scientific training and research background includes biochemistry, molecular biology, endocrinology, and academic drug discovery. He's spent the last several years in software as a scientific solutions consultant across various different industries, but with a very heavy focus on biopharma R&D. Larry will discuss HELM biopolymers in Signal Notebook workflows, and I will give him the driver seat. Thank you, Larry.
All right, thank you, Zev. Let me share my screen. Thanks for that introduction. And thanks, Lindsay, for the nice demonstration of utilizing the tools inside Signals. And for my portion, I just wanted to expand on the use cases of these modalities within workflows that, that might be common when you're working with them. So for my example, I've picked a modality of a peptide drug conjugate. So in this case, you have a targeting peptide that's targeted against some receptor. Typically, a lot of the peptides in this case are targeting cancerous cells with a linker and a payload. So not unlike antibody drug conjugates, of which there are several approved therapeutics on the market and a lot of development in that. But compared to antibody drug conjugates, there are some desirable properties of this. Much lower molecular weight, potentially better tissue penetration simply due to that molecular weight and size, and maybe tunability of the targeting peptide itself. Some potentially improved pharmacokinetics. And when you look at the synthesis and scale up of this, you could be talking about, solid phase synthesis and reactions as opposed to bioreactors, purifications, things of that nature with antibodies. Additionally, there's potentially some payload flexibility because of the size of the targeting peptide and not interfering with, steric hindrance of the antibody. So there's a lot of desirable properties here and a lot of potential for this. And I think with a lot of therapeutic modalities, there's a lot of academic research on it. And until there's a successful clinical candidate, you don't see a lot of players jumping in. But in this case, we do have at least one successful approved medicine in both Europe and the FDA in 2018, which is Lutathera from Novartis. And this is a targeting peptide that goes after the somatostatin receptor in a class of tumor cells, and specifically their payload is radioactive. So it's a really specific targeting of radiotherapy in essence. So this shows the power and potential of this platform. And one of the reasons I chose to use it is that it is a complex modality, right? The peptide could be represented by FASTA and in programs like, SnapGene or within our notebook, but the linkers and the payload represent potentially unique chemistry. So let's take a look at the workflow I'll go through here. And what I've decided to just show you some different facets of signals here is going through a QC check on some synthesized conjugated peptide drug conjugates here. And in the flow, what I'm going to go through is showing you the task request workflow in Signals Notebook that, okay, we have some available things that we'd like to run. I'll show you picking up those tasks and actually running the experiment. Through the course of that, I'll show you how everything is interconnected so that you can get the context you need through the course of running your own experiments and asking your own questions there. And I'll show you some of the other modalities in here as well. So we're very excited about HELM being a supported modality and expanding the capabilities of ChemDraw within Signals. But I'll show you some other things as I pull in a receptor protein for a binding study that's available in the platform as well, just so everybody's aware of all of the different ways we're covering this space. All right, so let me flip over to my Signals tenant, where I'll start with my login here and start on my Signals dashboard. So when I come to my Signals dashboard, you can customize your dashboard for certain settings. Included on mine is the ability to see that I have submitted peptide QC tasks. So these are essentially easy to build so that you can have these notifications right here when you're logging in, including your recent experiments and things like that. So because I see this on my dashboard, I know to navigate in the platform and come into my task dashboard here. And I've set up my task dashboard to be pre-filtered for the things that I'm interested in here. So you can see I have basically some filters around tasks that are submitted and go to a specific type. So if I go across this task, you can see it's a peptide QC. It has status of submitted. I can see the request link to that as well and all the other relevant data. I can, of course, clear out those filters and you can see all of the tasks on the dashboard and then simply add, any kind of filters you want with a quick type of search. So I can see all of those really quickly from my dashboard. Now, tasks are essentially a way of handing off work within the platform, right? It's giving you all of the context of the items that I require in the task, whether those are peptide drug conjugates in this point, inventory containers, samples, whatever the reference is, you can include it. And instead of emailing information around and pointing to links, it's captured all right here within the platform. So in this case, what I'm going to do to pick up this task is create an experiment around it. And you'll see I'll use one of my templates that's been built for this specific task. And you can see the description of it is for looking at the binding, the solubility, and serum half-life determination, all kind of QC tests that you might want to run before you do some deeper testing with these candidates going forward. I'll put this as my example QC run for today and select a notebook that I'm going to place this into, which is going to be my peptide drug conjugates, and I'll create the experiment. Now, once I've done that, what happens with this is essentially it creates a table in here. I'm going to drag that to the top in my template. You can see this is a new piece of my experiment, the task to do. And with the task to do here, this assigns all of the relevant information directly into my experiment. So that lets me track it and link it. And importantly, you can see, right, my experiment is linked. You can see the status of the task has changed. You can see the analyst is me. I've assigned it to myself in this case. So all of that context of picking it up comes through, and now someone's going to know, the creator of this will know that this task is in progress, actually, as you look across system notifications. And I used a template here, which is easily configurable to put in a host of tables that work throughout the platform. So you can do all of the context of your experiment in one place. Some of that is simply capturing experimental narrative, any variances, any setups that might be important. So I have some experimental notes here for one thing. And then I have a few tables in here. So one of these is a material table. So these will be the peptides I've tested here, which you can see right at the top. And if I wanted to pull in any additional context around these, I just have to enter the IDs into that, and it will pull up the names of these and any other configured fields that I've included in my registry, which is essentially here within the materials smart folder. So now you can see I have assigned IDs for all of these different peptide drug conjugates. You can see the HELM notation combined with the chemistry here for the conjugation. And in this experiment here, the kind of experiment that I've done, I've got a peptide IRGD that's specifically targeting and linked with a cleavable linker. And one of the things I mentioned is getting context on everything, right? When I pull that in, I can actually see the original experiments where these were synthesized. So if I wanted to go back to that original experiment, it's just a click away from the context, which I've opened up in a new tab here. So you can see my valine, citrulline, PABC linker kind of outlined here. This is generally cleavable with lysosomal enzymes when it's uptaken. There can be some cleavage within the serum as well, but generally you see the principle here, and this is a common linker used for antibody drug conjugates as well. So this would be the original synthesis, and like what Lindsay showed, I have an actual reaction here of conjugating the IRGD peptide itself to the linker, making that final product. And one of the things I wanted to show you in this, Lindsay showed you a reaction as well. It's a relatively newer feature. This IRG peptide is one of my reactants, and it is a registered material in my system, right? So it has a defined ID, but I actually used a new feature where you could do inventory decrements directly. So we've had this feature where you can add items into the reaction with a quick add feature here, which is how I added that in, but you can also do now container decrements. So if you have starting materials like these biopolymers or any of the linkers or things in inventory, it's possible to decrement that directly from a stoichiometry table and have all of that context with the actual containers you've selected on down the line. So this lets me come in and see, any associated QC data that went in with the synthesis before I run my own tests here. All right, so closing out that and returning to the context of this experiment here, let's continue down the line. We know the peptides we're going to use. We're going to specify, let's say, a protein we're going to use. And there's a few ways you can pull this in. So I showed you one way of interacting with inventory in the stoichiometry table. This here is a newer feature that is currently in beta. It's a nice one, though, because what it lets you do is look up an existing material by a container barcode. So you can see I can enter the ID that you've seen there, or I can scan a barcode. If I have a barcode reader that can scan into that table, you click into it, maybe scan a vial, enter in a barcode ID, and it will look up that material directly. So this material now is a protein itself. So it is an integrin receptor. That's what this IRG peptide is targeting. So a particular receptor candidate. This is what I'm going to use in the binding here. And you notice again, the barcode and the container ID have come directly in if you scan it. And if I were to specify that I want to use some amount of that in this experiment, I can do that as well. So that decrements directly from our inventory application, which is just a click away to see the container. I can see that the container is currently in use by me. So using those decrement features there automatically do that to connect you to inventory and update the amounts. And if I were to take a look at this particular protein, as you've seen some of the HELM biopolymers, we can see batches of this, containers available. You can see that amount I just took. I had 100 micrograms in each container. And if we look at the top level of this with the protein sequence, you can see some characteristics of it as well as the total container amount available in my inventory. Now, I mentioned we would showcase some of the other modalities here. So I am navigated now to my registry. I'm in the materials and I'm on a protein here. And this is one type of modality we capture within experiments and within our registry. So that would be a protein. This is obviously a more simplistic representation of larger molecules where you probably don't need the full chemical representation for something like a receptor protein, but you have full access to all of the information here in a sequence file. So this is just, a GenBank, a FASTA, a Snapgene type file loaded up and registered, given a defined ID, and connected into inventory. Along with this, if we go back to my particular batch here, we can see that this was made from an expression vector. So that also has a material ID, right? And is a DNA modality captured in here. So again, like everything else, we can capture within our library, plasmids. We're working on RNA as well, proteins, and you've seen HELM biopolymers. And essentially this toggle shows you all the different modalities that I have captured in here. And actually one of the nice things that we've done, so I have a biopolymer library specifically that has these types of peptides within them. This is essentially another chemistry library. So within the last few months, we've actually released the ability to configure as many chemically aware libraries as you would like. So you could now configure a library for peptides, oligos, conjugates, whatever you'd like to capture and separate those modalities. All right, so let's return to our experiment in our context here and some of the other tables that I have. Obviously, due to time, we're going to simplify some of the capture steps here, but I have a table that's configured in Signals Notebook to capture my SPR run here, capture an instrument that I'm using, and I can quickly enter any data related to that run. You notice with the template, my steps have already been set up, so you can pre-templatize that just to speed up data capture for your end users. But it also provides the structure around how you expect the data to be captured, or at minimum, what you're expecting to be captured here. So I have that captured along here, and then maybe I've run my experiment and I have some raw data that I would like to add into this. So I could just take from my computer locally all of my files that I have from somewhere and append those to the raw data section. So any of my SPR data, Excel, and also original files can come into this. I can see previews. I can work with this data if it's in a workable form, but importantly, I can keep it all attached into the context of the overall experiment. So now that that's all essentially done, the last step would be, entering in QC results here in a summarized table. You might structure this a little bit differently, but we've got all of our peptide IDs and all of the values in there where we're measuring properties of these peptide variants. And if you looked closely at the name, essentially what I did was modified the glycine at position 3 of the IRGD peptide to different glycine variants. So essentially testing how different glycine variants might affect different properties downstream. How soluble is it? What's the plasma stability? What's the binding properties of that by doing that? So looking for more desirable properties overall. So with all of that data entered in, right? The steps that you would typically take after that is, of course, potentially signing this and adding reviewers saying that it's done. But when you have a task as part of this, when it's integrated into that handoff of work, really you want to be able to complete the task at the end. So acknowledging that all of the data is done, that task status goes to completed, which I will see on the dashboard, which I will see on the original request for that 360-degree view. This is the actual request that started it all off. And I will also see, because I'm self-assigning and self-completing here, you'll see within my notifications and then within my email that I'm getting that information here too. So you can see I was assigned the task and you see the task was ultimately completed. So that lets you finish an experiment from end to end. Now, one of the other things I mentioned is in terms of finding context, right? You saw easy linkages here. So I was able to go to things that were readily linkable, like into my registry, into my inventory, into connected experiments throughout this process and see that it's any relevant information that I have. The other really excellent thing you have here for full context is search. So search can happen quickly in a variety of ways. You can have bookmark searches here, which I certainly have some bookmark searches. And I've gone to a number of different touch points in Signals here. So there's a number of ways to search across it. This includes, the new monomers that you see within there. The materials are registry essentially. And I just wanted to show you that if I was curious about other peptides that maybe use that linker, if I wanted to find some additional information, I could run a chemical search for something like this. So I could put that valine citrulline linker component in there and search for any related items in my library. So you can see all the peptide variants that I've created and quickly find out. So using the HELM features within drawing these, within reactions, registering these items, using them to create sample workflows and through tasks and requests are all essentially available. So really the features have been integrated throughout all of Signals completely. All right, so now that we've closed out that experiment, let me come back and just summarize what I went through here, just to show you the full context of the experiment. So we were able to capture complex biopolymers and conjugates, and Lindsay showed nicely how those could be drawn using the tooling there, using the libraries. We were able to register and track inventory, even decrement from inventory there. I showed you how the task can help you streamline handoffs between teams, between groups and individuals to keep the context within your experiments. And we can use search and the overall linkages that are natively built into Signals Notebook to help us get answers and context more quickly. All right, so with that, I think that concludes my part, and I can probably hand back to Zev to wrap us up.
Great. Thank you both, Lindsay and Larry, for your engaging demonstrations. I mean, I really think, not to continue the summary, but I think the HELM functionality in ChemDraw and showcasing those custom libraries with those abilities are really incredible, especially when I think about chemistry and biology folks being able to effectively communicate and collaborate working on the same biomolecules or same biopolymers. And one final thing I'll mention that I think, Larry, you did an excellent job just showcasing is like, how are these biomolecules being used in a true scientific use case, peptide drug conjugate example workflow. So that full end-to-end is really key and what we think makes Signals special. Before we get to questions, we will be posting a quick poll to gauge interest from our audience, and then we can open it up for Q&A. So the poll will be open now, and we'll leave that up for about 20 to 30 seconds. So go ahead and place your votes within there. And we appreciate everyone's voting. So we're about 9%, 10%. Give it 10 more seconds. I know it's some places it's early, some places it's late. So again, appreciate your participation. Can we get 50%? Almost there. Perfect. All right, we'll close that out. Really appreciate it. It looks like almost 50% want it now, and a couple people are still thinking about it. So we're excited to continue our conversations with that. All right. So we'll open us up for some questions. One of the questions that we received, which I think is really great, and this will be for Lindsay. We talked about it a little bit in our presentation, but how do you resolve a potential conflict of HELM representation that may arise from using one or more more than one library or predefined libraries? And is there a restriction on how many libraries that can be used? I'll answer the second question first. I believe there is no restriction. I think Larry mentioned that you can add as many libraries as you want. But Lindsay, maybe the first question on the potential conflict.
Yeah, so this is a really good question because it brings up a really good point. So you have the ability to add as many of those custom libraries as you want, and you also have those standard libraries available to you. So the way that it works, so if you are drawing, you can of course choose whatever library that you'd like. We don't actually force there to be unique monomers across all of the libraries, so no duplicates. So you can have essentially your own custom library that has all the monomers that you would use. And the way that you kind of resolve duplicates across other libraries is in the system configuration, the administrator actually chooses the search order of those libraries, meaning that if I had everything that I want and need in my own custom library, I can have Signal search that first if I'm pasting in a Helm string, for example, to pull that monomer definition back. When you're drawing, of course, you use whatever library you want, and there's not really a conflict there as long as you're drawing and using the definitions that you want. But from a search perspective, from a pasting a Helm string in, for example, the search order of those libraries that's set on the system configuration is what controls that.
Great. The next question is for Larry. What advice would you give organizations just starting to implement an ELN system and strategies for adopting HELM?
Yeah, that's one of those questions that the answer is it probably depends. But I think the big thing that we've seen, because we are in a beta right now, is identifying the users that can help you test this out. The users that want to leverage these workflows, and dedicate a little bit of training to those users that can then distribute the information out. I think having your engaged and expert users first using it and then spreading that in the organization is one good strategy, depending on the size of the organization. Yeah, I think that's generally what I've heard of our customers that are testing this out. It kind of starts with a handful of users testing, and then asking us some questions on the product team, a little bit of a feedback loop, and then rolling it out to a wider base.
Great, thank you. This question is for Lindsay. Will ChemScript support HELM?
That is a really great question. At this time, there is not a plan, but that certainly is not out of the question for the future.
Great. Similar to the question for Larry on advice for organizations, are there resources available for researchers to learn more about applying HELM for their workflows? Larry, to you.
So right now, if you're referring to Signals, we don't have dedicated documentation, or I should correct myself. I believe there's some documentation in our ChemDraw documentation online with Signals in terms of using the editor, things like that. But in terms of other documentation, it won't exist until we're in a full release. But if you're interested in it, I've been kind of updating some information for anyone that wants access to this beta feature or set of beta features. and providing that to anybody who's accessing it. So we do have some guidelines and information, and I've generally, Lindsay, myself, or both of us have been meeting with people to demo this and get them up and running. So if you would like information, we're happy to share that. We're also happy to have a brief meeting to help you get started.
And then another question for Lindsay. What are some of the challenges companies face when trying to standardize monomer libraries across potentially global R&D teams?
Yeah, I think, the first thing is that the HELM notation has tremendously helped in terms of standardization, right? But there's still really wide variability in how you actually define those monomers. And so one of the biggest challenges is ensuring that everybody within an organization is talking the same language. They're using the same monomers and the same definition behind that monomer symbol, right, that you see in that graphic representation. So, bringing this into Signals and centralizing those library really ensures that everybody is actually talking the same language, right? Having a central truth to those libraries is going to start to ease some of that challenge that has been out there and being dealt with kind of within individual organizations, right? Trying to save libraries locally across an entire organization is simply not sustainable, right?
Sure. Quick follow-up question to that, I guess maybe both for you, Lindsay and Larry. Are there public HELM monomer libraries available to help new users get started with the polymer or biopolymer design?
Yes, there are. So in Signals today, we actually have standard available to users, a natural monomer library, also a library that the Pistoia Alliance actually curates and puts out. So those are there. The Pistoia Alliance has a public open access database for the monomers that they actually have curated themselves, but we also offer that in Signals as well.
Great. This is a question for me because I'm always curious between the biologists and the chemists. I know sometimes people can be at odds, especially when, it can be frustrating if there's not a tool to kind of easily communicate what we're working on and together. How do you think this will change maybe the relationship between teams, chemistry and biology that are working within these biopolymers to be successful? Larry, to you.
Sorry, Zev, my audio split a little bit there. I missed the
That's
last
okay.
half of your question.
Oh yeah, just was curious between sort of biology teams and chemistry teams who are now able to talk the same language. How do you think this will change the way they communicate and work together when previously maybe was much more challenging?
Yeah, that's a good one. The thing that I've heard prior to having these capabilities in Signals, or even, frankly, sometimes people weren't aware that we had some of these capabilities in ChemDraw that Lindsay showed have been around since 2017. What I found is that people have been sort of creating their own internal language to represent these things because the tools for design haven't exactly supported it. So they've kind of been making their own monomer libraries, if you will, with like shorthand text, which works with a small group, but in a wider organization means the fidelity of what you're making can go down quite a bit. It's like a real challenge. And you see this stuff, like I'm a biologist, so I saw this stuff all the time when you order, say, oligos from IDT, right? They have shorthand so you can upload an Excel spreadsheet to get modified nucleotides and things like that, which is great for the common things for oligos and the like. But these conjugates, like what Lindsay drew, what I was showing for peptide drug conjugates, are way more complex. So capturing everything that's part of it is just much more standardized. Like you really know what's been designed, what's in your material, and then that's essential. So for the chemist designing it and handing it off to like someone doing QC, like I was showing it, that really helps make sure you know what you've got.
Perfect. Well, that's all the time we have for questions. Let me close this out here. Thank you for everyone joining us today for our 2023 Signals Notebook Quarterly Connect community series focusing on HELM and the Signals Notebook. Just as a quick reminder, we will have our next session on November 29th. We hope you can join us. Reminder that everyone who has attended this presentation, you are already registered, so no need to re-register. And again, if you want in between sessions, you can stay up to date on our Signals Notebook new features and functionality via What's New section on the website and email communications. Thank you again for our speakers for sharing great insights. And we will address any pending questions in the upcoming days and share the presentation. Appreciate everyone's time. And that is all. Thank you so much. Bye, everyone.